Imagine a future where, genetic diseases aren't just managed, but actually corrected, right down at the DNA level. And think about the most complex biological experiments, things that used to take years, maybe being designed almost.
Well, on demand. Welcome to the deep dive. Today, we're
Hmm.
really pulling back the curtain on how artificial intelligence is rapidly transforming genome editing, specifically CRISPR technology.
Yeah, and for this deep dive, we've dug into leading research papers,
Mhm.
followed lively discussions in the scientific community, looked at expert analyses. Our goal is basically to bring you the most important nuggets of knowledge from this incredible intersection. Think of it as a shortcut to really getting up to speed on this field. Exactly. Our mission today is to unpack the opportunities, which are huge, the cutting edge tools, and also the really critical ethical questions around this powerful mix of AI and biology. We want you to understand how these technologies aren't just theory anymore, they're actually shaping healthcare and science right now. So let's just jump right in. Okay, so when we talk about editing the blueprint of life itself, what exactly is genome editing at its core?
Right. So at its core, genome editing is really about making precise changes to DNA sequences inside living cells. This capability has completely transformed how we study genes and maybe more importantly, how we develop therapies for, well, countless diseases. It's literally fixing or changing the genetic code.
And when people think about precise gene editing, usually one name pops up immediately: CRISPR-Cas9. Why do they call it molecular scissors? How does that work?
Yeah, CRISPR-Cas9 is definitely the most common, the most versatile tool we have right now. And molecular scissors is a great analogy because of how it works. You have a protein, Cas9, usually that's the scissor part, and it uses a little piece of RNA called a guide RNA or gRNA to find a very specific spot on the DNA. Think of that gRNA like a GPS coordinate, guiding the scissors precisely where to cut. Once it finds the target, the Cas protein cuts the DNA. Then the cell's own repair systems kick in, and they can insert, delete, or even replace bits of DNA right there.
Okay, so CRISPR gave us this fundamental cut and paste, but technology never stands still, right? How has it evolved? What are these newer, more specialized approaches?
That's a really good question, because the field moves incredibly fast. Beyond that classic CRISPR-Cas9 cut, we now have more sophisticated variations, like base editing, BDD. This lets you change just a single letter in the DNA code without making that double-strand break that cut across both strands of the DNA, which is a big plus for precision and safety. Then there's prime editing, or PED. This one is incredibly versatile. It's more like a DNA search and replace function. It can make more complex changes, insertions, deletions, swapping out sequences. And a newer area is epigenome editing, EpiGED. This lets us tweak how genes are expressed, turning them up or down without actually changing the underlying DNA sequence at all. Each of these has, specific advantages for making really precise programmable changes, often avoiding reliance on those complex, sometimes messy natural repair pathways.
So, with all this incredible precision, what does it mean for healthcare? What's the big promise of CRISPR for treating actual diseases?
Oh, the promise is immense, truly. CRISPR can be used to go after disease-causing mutations directly, things like mutations in tumor suppressor genes, or genes linked to heart disease. It can be used to knock out faulty genes, think sickle cell anemia or cystic fibrosis, or even insert new functional genes. We're seeing it being developed to target genes involved in neurodegenerative diseases like Alzheimer's and Huntington's, and even creating cells that are resistant to viruses like HIV and hepatitis B. The potential applications are just vast.
And like you said, it's not just theoretical anymore. We're seeing real progress in clinical trials. In fact, wasn't the very first gene editing therapy just approved?
That's right, a huge milestone.
Hmm.
Both the FDA in the US and the UK government recently approved the very first CRISPR-based gene editing therapy.
Mhm.
So this is moving beyond the lab bench. It's becoming a clinical reality, offering real hope for patients with certain genetic conditions. It's a massive step forward.
Okay, here's where it gets, I think, really interesting. Bringing AI into this whole picture. AI isn't just like a helpful add-on here. People are calling it the ultimate co-pilot for gene editing. Some even say it's imperative. Why is AI so crucial?
You've really hit on a key point there. AI isn't just a nice to have, it's becoming fundamental. It absolutely holds the promise to revolutionize healthcare by making gene editing much more precise, way more efficient, and maybe even more affordable in the long run. Think about the sheer amount of data we get from modern biology. These multi-omics technologies looking at genes, RNA, proteins, DNA modifications all at once. It's generating truly big data, mountains of it. AI is basically essential to analyze all that, to interpret it, to find the patterns. It can help predict, how likely a specific edit is to work based on past results. It just makes the whole process smarter.
So AI helps make sense of all that data. But how does it help do the editing? Specifically designing those guide RNAs, the gRNAs, that tell CRISPR where to go. Getting that right seems absolutely critical.
It is absolutely critical. Designing effective gRNAs is vital for two things: efficiency, making sure the edit actually happens, and specificity, making sure it only happens where you want it to, avoiding off-target effects.
Yeah.
And this is where AI models are really indispensable. Tools like Deep CRISPR or CRISTA, these are sophisticated AI models designed to predict the best gRNAs. They look at tons of factors: the DNA sequence around the target, the type of Cas protein used, the specific change you want to make, and they calculate scores for how well it should work on target and how likely it is to hit off targets. They learn from millions of examples.
And I've heard about even more advanced AI models coming online now. What makes them different?
Yeah, the pace is just relentless. We've got models like Tiger and CRISPR. These use really powerful AI techniques like convolutional neural networks, CNNs, and deep learning. They're getting even better at predicting both on-target success and, crucially, those unwanted off-target cuts. And then there's a tool called Pick CRISPR, which is quite interesting. It uniquely combines the DNA sequence info with physical properties of the genome. Things like how tightly packed the DNA is in that region, or chemical tags like DNA methylation. This gives it, theoretically, an even better understanding of where those off-target cuts might happen, aiming for even safer edits.
Wow, that sounds incredibly detailed. So how does AI help with those newer types of editing? We mentioned base editing and prime editing.
Right, so for base editing, BED, where you're changing single letters, AI models like BEDICT and DeepAB are proving really good at predicting how efficient an edit will be and what the outcome might look like. This directly helps in designing strategies to correct specific genetic mutations. And what's really exciting, I think, is using AI tools like AlphaFold 2, which is famous for predicting protein structures, to actually design new base editors from the ground up, engineering them with specific desirable features. It's like AI-driven tool design.
In prime editing, that sounds even more complex. Designing those special guide RNAs, the PEG RNAs, must be a challenge. How's AI helping there?
It definitely is more complex, yes. Prime editing, PED, uses these longer, more intricate guides called PEG RNAs. But again, AI is stepping up. Tools like EasyPrime and Prime Design help researchers design these PEG RNAs and predict how well they'll work. There's even a deep learning model called Predix that tries to forecast the outcomes and rates of prime editing. It even considers things like the activity of certain DNA repair proteins in the cell, like TREX1 and TREX2. Because these repair proteins, they're like the cell's cleanup crew, and sometimes they can interfere with the prime editing process. So AI helps us anticipate and maybe work around that.
Okay, and what about epigenome editing, EpiGED? That's newer territory, right? Is AI playing a role there yet?
It's definitely an emerging area for AI application. It's a bit trickier because epigenetic marks can be more dynamic than the DNA sequence itself, but tools are starting to appear. EpicSDL, for example, is being developed to predict sgRNA activity specifically for epigenome editing. The goal there is to regulate gene expression precisely, turn genes on or off without ever touching the DNA code. The potential for fine-tuning is huge.
This all really feels like it's converging towards truly personalized medicine. How does AI connect the dots between the lab work and treating an individual patient?
That's exactly where it's headed. The combination of AI, genome editing, and precision medicine allows for treatments tailored to a person's unique genetic makeup. AI can sift through a patient's genomic data, identify specific mutations or variations linked to diseases like cancer, diabetes, Alzheimer's. We have machine learning tools like AlphaMasense, for instance, that predict whether a specific protein variant is likely to cause disease. Deep neural networks are helping identify potential gene targets for rare diseases. Then CRISPR can potentially be used to make the precise correction needed for that individual. AI doesn't just make this possible, it makes it scalable. It helps democratize that deep genomic insight.
And what about actually getting the CRISPR tools into the right cells in the body? That delivery problem has always seemed like a major roadblock.
It absolutely has been and still is a major hurdle. But AI is proving really valuable here too. It's being used to help optimize the delivery methods for CRISPR components, whether that's delivering them as mRNA, DNA, or as ready-made protein RNA complexes. AI can help design or refine the delivery vehicles like lipid nanoparticles or viral vectors, tailoring them for specific tissues or cell types. The idea is to get the cargo exactly where it needs to go, safely and effectively. It's a tough engineering challenge where AI's predictive power can make a real difference.
So we've seen AI as this brilliant co-pilot fine-tuning all these different steps. But what if AI could actually run the whole show, design a complex experiment from start to finish? And that brings us to something that sounds pretty revolutionary, CRISPR GPT. An AI agent specifically built for gene editing research.
Exactly. CRISPR GPT is a specialized large language model agent, an LLM agent. It came out of research involving Stanford, Princeton, and Google DeepMind. It's designed specifically for automating gene editing experiment design. And the real insight here isn't just the automation itself, it's that CRISPR GPT potentially shifts the bottleneck in scientific discovery away from laborious human effort and deep individual expertise towards the speed of computational design. That could fundamentally reshape how we do biology research. It streamlines these incredibly complex experimental designs by integrating the power of LLMs with very specific biological knowledge, reasoning capabilities, and computational tools.
That sounds incredibly powerful. How does a researcher actually use it? Is it just like asking a question and getting an answer?
It's more interactive than that and flexible.
Wow.
CRISPR GPT offers different ways to interact, depending on the user's needs. There's the meta mode. This is great for beginners. It gives step-by-step guidance through predefined tasks, choosing the right CRISPR system, designing the guides, even planning the data analysis. It's like having an expert mentor walking you through it.
Okay, makes sense for beginners. What about experienced researchers who know what they want to do but maybe want help planning the specifics?
For them, there's the auto mode. This allows for more freestyle requests. An advanced researcher can describe the experiment they want to run. The AI planner inside CRISPR GPT breaks that down into a customized workflow, figures out the tasks, manages the dependencies, and executes them using various computational tools. It can even fill in missing details and explain why it made certain choices, which is pretty neat. And then there's a Q&A mode for quick scientific questions. This mode pulls information together from a specially fine-tuned model called CRISPR Llama 3, plus peer-reviewed papers and general LLMs to give a comprehensive answer.
CRISPR Llama 3, that sounds specific. How did they train that? Where did it get its specialized CRISPR knowledge?
Yeah, that's a key part. CRISPR Llama 3 was trained on a dataset of real scientific discussions from a public Google group focused entirely on CRISPR gene editing. This means it's learned the language, the practical problems, the nuances, the common pitfalls that researchers actually discuss when they're doing this work in the lab. That gives it a much deeper, more practical understanding than a general LLM would have.
Okay, the concept is amazing, but the proof is in the pudding, right? Has CRISPR GPT actually been used to successfully run experiments in a lab?
It has, and the validation results are pretty compelling. The researchers had junior scientists, people who were not experts in gene editing, use CRISPR GPT to design and carry out complex experiments. And reportedly, they were successful on their very first attempt, which really speaks volumes about the AI's ability to guide the process effectively.
Wow, first attempt on complex experiments. What kind of experiments are we talking about?
Well, in one case, a junior researcher used it to design and perform a multi-gene knockout in human lung cancer cells. They targeted 4 different disease-related genes simultaneously: TGFR1, SNAI1, BEX-ANZO, and BCL2L1. And they achieved around 80% editing efficiency, which is quite good, especially for multiple targets. CRISPR GPT guided them through literally every step, selecting the best CRISPR system for hitting multiple genes. They used something called ASCO12A, choosing the delivery method, lentiviral transduction, designing the gRNAs, generating the lab protocols, and even analyzing the sequencing data afterwards using tools like Primer3 and CRISPR-SO2 to confirm the edits worked and check the biological impact.
That's impressive. Was there another example showing its versatility?
Yes, absolutely. Another junior researcher used CRISPR GPT to perform epigenetic editing. The goal was to activate 2 specific genes, NCR3LG1 and CACAM1, in a human melanoma cell line. Again, guided entirely by CRISPR GPT, they achieved high activation efficiency up to about 90% for one of the genes. This shows the AI isn't just limited to cutting DNA. It can handle these more nuanced epigenetic modifications too, precisely tuning gene activity.
So what's the magic behind the scenes? What makes CRISPR GPT work so well? What's the secret sauce?
Well, it uses a pretty sophisticated setup called a multi-agent architecture. Basically, different AI components specialize in different tasks. There's a user proxy that understands your request, an LLM planner that figures out the experimental steps, a task executor that runs the computations, and a tool provider that gives access to necessary bioinformatics tools. They all work together. It also uses advanced AI techniques like chain-of-thought reasoning, where it breaks down the problem logically, step by step, and retrieval augmented generation, or RAG. RAG lets it pull in relevant, up-to-date information from scientific databases and literature as it plans. This whole combination allows it to significantly outperform general-purpose LLMs on tasks specific to gene editing, like planning experiments or designing those crucial gRNAs.
This is all incredibly powerful, but this convergence, AI guiding our ability to edit life's code, it inevitably raises some profound questions, doesn't it? Especially this idea of optimizing biology.
It really does. There's this influential way of thinking in modern biology, especially spurred by genomics and CRISPR, that views living systems almost like machines.
Hmm.
Machines that can be programmed and optimized. We talk about mitochondria as powerhouses, senescence as molecular motors, engineered microbes as tiny factories. Now, this machine analogy is useful. It helps simplify complex ideas, but it could also foster a mindset of trying to optimize human biology beyond just treating diseases. And that's where the lines can get really blurry between health and enhancement.
And that blurring leads directly to a major ethical fault line, the difference between somatic and germline editing. Can you break that down? Why is one so much more controversial?
Certainly. Somatic editing means making changes to the DNA in regular body cells of an adult or child to treat a disease they already have, like editing blood stem cells for sickle cell disease. These changes affect only that individual. They aren't passed on to their children. So, while still needing careful ethical review, it's generally seen as less controversial. Many therapies and trials now are somatic. Germline editing is different. This involves editing DNA in sperm, eggs, or embryos. The key difference is that these changes would be heritable, passed down through generations. The idea of making permanent, intentional changes to the human gene pool raises huge ethical questions about unforeseen long-term consequences, fairness, and really what it means to intentionally reshape our species. That's why it's far more contentious.
And what's the situation in the US right now regarding germline editing? Are there specific restrictions?
Yes. In the U.S., there are significant restrictions. For example, the National Institutes of Health, the NIH, is prohibited from funding research where human embryos are created or destroyed specifically for research. And currently, the FDA is effectively barred from reviewing applications for clinical trials involving heritable germline modifications. This creates pretty strong barriers to this kind of research, especially medical research in the U.S. The debate often circles around enhancement, trying to improve traits beyond normal versus strictly disease prevention. And that's a line that AI, being essentially value-free, won't draw for us. It just executes instructions.
How does the public feel about this? And does it very much around the world?
It does vary. In the U.S., generally, polls show support for somatic editing to cure serious diseases. But there's much more caution, even disapproval, when it comes to enhancement or germline editing. Some studies suggest this is partly linked to lower average scientific literacy about the details. If you contrast that with, say, China, there's been incredibly rapid progress in both genomics and AI, and public surveys there have sometimes shown a more positive or accepting view towards research involving human embryo editing, at least for disease prevention. The cultural context and regulatory environment definitely play a big role.
And those differing regulations and public views could lead to situations like medical tourism, right? People traveling for procedures they can't get at home.
That's a very real concern, yes. That patients might travel to countries with perhaps looser regulations to access gene editing procedures that are restricted or banned in their own countries. We've seen precedents for this with other advanced reproductive technologies. Mitochondrial donation, for example, was performed in places like Mexico for patients from countries where it wasn't legally permitted at the time. It highlights the need for some level of international discussion and coordination, which is obviously very difficult.
So, despite all the amazing potential, we still need to be aware of the challenges and the necessary safeguards. What are some current limitations of AI in this space?
Absolutely. AI in gene editing isn't perfect. Models can still struggle with rare biological situations because they're trained on data. And data for rare cases is, well, rare. So their predictions might be less reliable there. There's also a need to train AI for designing guides beyond just human and mouse genomes to cover the huge diversity of organisms researchers work on. And a persistent challenge is the black box problem with some complex AI models. It can be hard to know exactly why an AI made a specific prediction or recommendation. In a clinical setting, that lack of explainability is a real hurdle for risk assessment and trust. If something goes wrong, you need to understand the reasoning.
It sounds like the developers are thinking about ethics from the start, though, building in safeguards.
Yes, and that's crucial. Responsible developers, like those behind CRISPR GPT, are building in ethical safeguards. For instance, ensuring patient data privacy is paramount. things like hypercompliance, filtering out potentially identifiable genomic sequences before they might be sent to a general LLM, and critically, including explicit warnings and potentially even blocks against using the tool to design experiments for human heritable germline editing, trying to prevent misuse and align with international guidelines. That kind of built-in ethical consideration is vital.
Looking ahead then, what's next? What's on the horizon for AI and gene editing?
Well, one exciting direction is tighter integration. Imagine AI-guided design seamlessly connected to automated robotic lab platforms. This could massively accelerate the pace of discovery, basically running experiments 24/7 with minimal human intervention. We'll also see AI models being developed and refined to support the growing toolkit of gene editing technologies beyond just the basic CRISPR-Cas9, like base and prime editors, and maybe others yet to come. And fundamentally, there's ongoing critical research needed into AI model explainability, making those black boxes more transparent. Also, better ways to select the most relevant biological features for training AI and building more comprehensive, centralized databases for CRISPR experiments. All of this is key to ensuring these powerful tools are used safely and responsibly.
So this deep dive really highlights the incredible synergy here. AI is making gene editing sharper, faster, more accessible, from designing tiny molecular guides to planning whole experiments and tailoring treatments. It lets us seriously imagine a future where AI-guided gene editing could unlock solutions for health problems we can barely tackle today.
But it also forces us to confront those big questions. As AI helps us get better and better at optimizing biology, maybe even human biology, where exactly do we draw that line, the line between healing and fundamentally altering ourselves?
Yeah, what does an optimized human even look like? And maybe the tougher question, who gets to decide? As you listen to this, what stands out to you as the most profound implication of this powerful convergence between intelligence and the code of life?